So what was the Bristol GDNF trial? In January 2010, while the gene therapy clinical trials were being conducted for neurotrophic factors, Amgen licensed the rights to GDNF to a company called MedGenesis. And the folks at MedGenesis were very keen to see another clinical trial in Parkinson's. Together with the Cure Parkinson's Trust and Bryn Williams, Professor Gill approached Parkinson's UK and proposed conducting another Phase 2 double-blind clinical study of GDNF in individuals with advanced Parkinson's. Parkinson's UK agreed that it was a good idea and also agreed to fund the bulk of the study. The new study began in 2012, and it involved a revolutionary new method of delivering the treatment, which involved tubes being implanted in the brain with pinpoint precision thanks to the assistance of robotics, a system developed by Professor Gill and a company called Renishaw. These tubes connected with another tube which ran to a small port which was placed just behind the ear. Source: Medium, a very good article from Parkinson's UK discussing the study. And it was via this port that the GDNF, or placebo, treatment was delivered to the participants in the study. A participant receiving treatment. Source: Parkinson's UK. I am often referring to added value being an important part of any future clinical trials, and this delivery system is a fantastic example of this. This same technology is now being used in the ongoing CDNF clinical trial for Parkinson's being conducted in Finland, Sweden, more on this below, as well as multiple oncology clinical trials. Regards of the results of the GDNF trial, the output of the study has already had tremendous benefits, added value, for the wider medical community. Sounds very interesting. So the results of the study were published today? Yes, in the prestigious journal, Brain. Title: Randomized Trial of Intermittent Intraputaminal Glial Cell Line-Derived Neurotrophic Factor in Parkinson's Disease. Authors: The researchers began by screening individuals to identify those appropriate for the study. Between October 2012 and April 2015, 196 individuals from throughout the UK were screened. Of these, 41 individuals were selected to take part in the study. The investigators began the study with a small pilot group of participants, 6 people, to assess the safety of the surgical technique and the administration of the GDNF. Once optimized, the remaining 35 participants were given the surgical procedure. All of the participants were then randomized and treated for 40 weeks with either GDNF or the placebo treatment. Click here to read more about the details of the trial. The primary endpoint of this trial was the percentage of change from the baseline measure in the practically defined off-state UPDRS motor score, Part 3, after 40 weeks of double-blind treatment. This means how the degree of improvement in participants when they are not on their medication, between the start of the study and the end of the study. Clinical assessments were conducted at the baseline, 8 weeks, 16 weeks, 24 weeks, 32 weeks, and 40 weeks time points. The secondary endpoints of the study included the percentage of change between baseline and the weeks 40 time point in UPDRS motor score in the on-state, that is, the motor performance of participants while on medication. The percentage of change from baseline in UPDRS activities of daily living scores in the off and on state, the UPDRS parts 1 and IV scores, the change between baseline to the week 40 time point in Parkinson's diary ratings. In addition to these, numerous supplemental endpoints were also measured, including fluorodopa brain imaging. The imaging was conducted at baseline, before treatment, and again at the 40 weeks time points. The results of the study suggest that there was no difference in off or on performance at the 40-week time point between the GDNF and placebo-treated group. As you can see in the graph below, both groups began improving soon after treatment began, and they both continued to do so across the 40 weeks of the study. A decrease in UPDRS score suggests an improvement in the motor performance of the participants, and both the red line, GDNF treatment, and the blue line, placebo group, on the graph have a downward trajectory. Admittedly, the red line is lower than the blue line, but this never gets statistically significant. How do we explain this? There may be a difference of opinions here, but I would lean towards a placebo response. What is a placebo response? A placebo response occurs when one exhibits a reaction to a treatment, for which there is no pharmacological reason. There is no biological reason why the placebo treatment caused the control group to improve, but they did. In addition, none of the secondary endpoints exhibit any significant differences between the two groups. This is really bad, right? It is unfortunate, but it is not the end of the world. More importantly, there's some very interesting findings in the overall results. Such as the responders. Every drug trial has individuals who respond better than anyone else in the study, and this study was no different. Some members of the GDNF treated group responded better than others to the treatment. But crucially, this responder difference points towards a difference between the GDNF and placebo group. You see, when the researchers looked at number of participants who achieved a better than 10-point improvement on the OFF-UPDRS score, there were 9 who experienced this in the GDNF group, but none in the placebo-treated group. While there were many individuals who had a placebo response in the control placebo group, none of them had a response as big as the GDNF responders. And you can see this as the graphs below. On the left, GDNF treated group, there are 9 red bars beyond the greater than 10-point responder line, but on the right, blue side there are none. Source, brain. Interesting. Anything else of interest? Yeah, the brain imaging results. Have a look at the image below. Source, brain. Each of the 4 green circles represents a bird's eye view of the brain. The red, yellow regions are the putamen on each side of the brain, and the greater the intensity of red coloration, the greater the level of dopamine activity. Now, if you look at the 2 brains in the top row, these are images from the same individual, baseline image on the left and 40-week image on the right. Note the subtle reduction in red coloration in the right 40 weeks image compared to the left baseline image, particularly around the bottom tips of the putamen. This loss of red coloration suggests a reduction in dopamine activity. The images in the top row came from an individual who was treated with the placebo during the study. Now shift your attention to the bottom row of brain images, and note the increase in red coloration in the right-hand image, the 40-week time point. I hope you will agree that compared to the baseline image left, there is a lot more red coloration on the 40 weeks right image. This difference suggests an increase in dopamine activity. What could the brain imaging results mean? It suggests that the GDNF was having a biological effect in the brain, the treatment was doing something. Exactly what that something is still needs to be determined. There are in effect 3 possible explanations. 1. There is an increase in dopamine turnover in the remaining dopamine fibers. 2. There is an increase in the number of dopamine fibers. 3. There is an increase in other types of neuronal fibers which can take up levodopa. All 3 explanations are possible. But we currently have no means of testing which is the answer, beyond repeating the experiment with a larger range of brain imaging assessments. But I hope you will agree that this is a very interesting part of the results and it certainly deserves further investigation. Interesting. Anything else? One other really interesting detail in the results: no antibodies. Across both the double-blind and open-label parts of the study, the researchers never detected any antibodies against GDNF in the analyzed blood of participants at any time during this study. Note, after the 40 weeks of double-blind study, there was another 40 weeks of open-label assessment in which all of the participants received GDNF. Click here to read the report on that phase of the study. So what happens next? Well, this does not appear to be the end of GDNF or neurotrophic therapy approaches for Parkinson's. There are a lot of ongoing discussion regarding whether there should be another trial to follow up the responder data and the brain imaging results. There are questions being asked as to whether the dose of GDNF was high enough to have an effect. The dose used was 3.5-fold smaller than in the historic continuous dosing studies, 120 micrograms versus 420 micrograms, for safety reasons based on the neurodegenerative results from primates. In addition, some folks are asking if the study was long enough. And while these discussions regarding GDNF continue, we should get some news soon about another neurotrophic factor clinical study which is using the same delivery system as the Bristol GDNF study. The cerebral dopamine neurotrophic factor (CDNF) clinical trial being conducted in Finland and Sweden is scheduled to finish this year, and we will hopefully get some news about that study. This study is being run by the biotech firm Herantis. The Phase 1-2 study is evaluating the safety and tolerability of CDNF in people with Parkinson's. Similar to the Phase 2 GDNF study, the drug is injected directly into the brain using an implanted cannular system. 1/3 of the participants have received monthly infusions of placebo and 2/3 of the participants have received monthly infusions of either mid or high doses of CDNF for 6 months. Click here to read more about this trial. An extension study of this trial has recently been initiated. Click here to read more about that study. The top-line results from this study will not be available until next year, but the safety, tolerability data from this trial are going to be presented the Alzheimer's Parkinson's 2019 meeting in March. It will be interesting to see what those results suggest. In addition to this study, there is a gene therapy GDNF clinical trial starting in California. This study was originally supposed to be conducted on the East Coast of the US, but it has been shifted to the West Coast, and it will hopefully be starting in the summer. This study will involve an AAV virus being injected into the brain, which will infect cells with the DNA required for the production of GDNF, causing cells in a particular region of the brain to start producing GDNF. Click here to learn more about this trial. We will be keeping an ear to the ground for information relating to this study. And on top of this, there is also a company called GeneCode, which is developing orally delivered, small molecules that mimic GDNF and trigger neurotrophic signaling in dopaminergic neurons. Source. It will be interesting to see what happens with this company. Phew, long post. So summing up. What does it all mean? Not just yet. There is something else we really need to discuss. There is no way of avoiding the fact that there was an enormous placebo response in the control, placebo-treated group in this study. As I mentioned above, a placebo response occurs when one exhibits a reaction to a treatment for which there is no pharmacological reason. There was no biological reason why the placebo-treated group should have improved, but they did. Some folks might suggest that the surgical procedure instills a strong chance of placebo effect. Others will point towards the excellent and dedicated clinical care at a single research site, which was committed to maximizing patient retention, possibly aiding this situation. While I am open to theories or ideas as to what could have caused the placebo response, I am biased towards a more human answer. This trial started with some mighty big expectations on the part of everyone involved, both the participants and the coordinators. And this potential for a placebo response deeply concerns me as we start to put further resources towards large clinical trials for treatments that the community already has high expectations for. Think, exenatide. Placebo response is acknowledged as a problem in Parkinson's. Click here to read an old SOPD post about this, and the Bristol GDNF study provides a fine example of it. We need to have a careful think about managing expectations going forward. So what does it all mean? I hope I will be forgiven for using the roller coaster analogy in this post, but I have watched the GDNF saga from the sidelines since starting my PhD in 2002. The lab I conducted my doctoral research in was one of many that produced some of the early preclinical GDNF data, so I was surrounded by the buzz of excitement about this amazing new compound. I have read The Monkeys in the Middle book by Nick Nelson, a very good, but slightly dated review of the GDNF story, and I have followed every subsequent twist and turn of this tale. I like to think that have provided a fair overview of the GDNF story and description of the latest clinical trial results in this post, and I am happy to be corrected on any details. But please understand, there is a terrible sense of having been to this rodeo before. Of having watched the roller coaster do the loop several times. God Almighty, it's déjà vu all over again. There are going to be lots of voices in the aftermath of these results being published, and my hope is that unlike many previous unsuccessful clinical trials which have been swept under the rug and forgotten, 40% of Parkinson's clinical studies do not publish their results. Click here for more on that rather damning statistic. I hope that the Bristol GDNF clinical trial will be the trigger for change. An acknowledgement by the entire community that our clinical trial process needs to be carefully reconsidered and adapted. A very sober analysis of the Bristol GDNF study is required. It should involve voices from all sides of the clinical trial process. Patients, clinicians, everyone. A constructive conversation about how we can improve things and do better. Please let us learn from this experience, and use that knowledge to plan for better, and ideally more successful, future clinical trials.